Okay, we'll talk a little bit. They kind of look very similar. And for a very long time, they were classified in the same kind of kingdom. But then we noticed some differences. Yeah.
Hydras?
No, hydras aren't actually the eukaryotic. So we'll have hydras later. Good try. Yeah. Protists are eukaryotic too. So yeah, but there are some.
Oh.
Yeah.
Protozoa.
Protozoans are eukaryotic too.
Oh.
They're part of the protist group, which is our next. Okay, don't worry. Okay. Okay. So, one of the reasons that prokaryotes are of interest is because they've been around for a very long time, and evolutionary speaking, they haven't had a whole lot of changes. So that kind of indicates that they've been like a very successful group of organisms, and their ability to adapt to reproduce, survive harsh environments has contributed to their success over kind of since the beginning of life. Okay. So kind of going over some characteristics, we already talked about this, most of this, prokaryotic cells and eukaryotic cells, that was just as a review. In general, they have very little organelle. No nucleus is the major difference. And then there's also much smaller in size, okay? And reproduction can be asexual or sexual, but again, very different methods for eukaryotic cells. A lot of them are unicellular, okay? These are single cells, but you can have colonies which are single cells that join together they live together. Some evidence suggests that maybe that is kind of the beginning of multicellular organisms, that individual cells can actually survive and coordinate with each other. And they have their cell membrane, cell wall, they have DNA, genetic material, and they have ribosomes to make protein, okay, because they do have a membrane, right? So they need to make some proteins for that. Cytoplasm, and a lot of them have mobility structures like flagella, cilia, to help them propel through usually a liquid medium. Okay. So we're going to be growing some bacteria, but you're probably not going to see all of them, because we're going to probably see colonies of bacteria because they're quite tiny and small, so light microscopes is not going to be, in terms of magnification, not be high enough for you to observe those characteristics. But hopefully you'll get to see some colonies and be able to identify different types of bacteria based on the colors and colony size and things like that. All right. Classification can be a little bit tricky. We're not going to go into a lot of detail in terms of various groups in bacteria classification. So we're just going to look at major characteristics. Okay, so that's kind of all you need to know is some traits that may be used to divide bacteria into different groups. Okay, a lot of it now is obviously through genetic sequencing. Okay, so looking at which alleles are shared by individuals. But we can look at, there are three major shapes. Okay, and again, you have this on your sheet. We have kind of spherical ones, aclossi, and then rod shaped, which are the bacilli, and then you have this rurella, which is like a kind of like a squiggly, kind of elongated structure. Okay, and so bases off of that, the kind of the basic shape, you can have colonies of organisms. So you can have a couple or form a chain, and these, again, they would be a colony of individual cells that live together, and we really consider them as one organism. So shape can help us categorize various strains of bacteria. We can also use something called grand staining. Okay, so this has to do with the cell wall composition that is found in particular strains that you're looking at. So we can use particular dyes that will stain these the membrane and the wall of the bacteria differently, and based off of the color that we see under the observation under a microscope, we can say, hey, this is this type of bacteria is gram-positive versus gram-negative. And so the big difference between the two groups is the actual composition. So cell membrane usually is made of lipids, okay, and protein molecules inserted in the lipids. What's kind of unique about prokaryotes, bacterial prokaryotes, is that they have a layer of carbohydrate that kind of composes kind of a mesh-like structure between the lipid membranes, okay, and that is called peptoglycan. Okay. And so some groups have lots of peptoglycan, have layers of them, okay, and so they will sting a little bit differently because that peptoglycan is going to grab onto the color dye. Versus others have very little, and so they're going to be a slightly different color when you put the dye on. So you can use that kind of strategy to help identify the different strains, and that will tell you a little bit about the composition of the cell membrane, cell wall of the organism. We can also kind of group them based on the way they feed. So heterotrophs versus autotrophs. And so what's that again? Yes?
Autotrophs can make their own food and heterotrophs need to consume others.
So what are some ways that autotrophs can make their food? Photosynthesis, right? So we call it photoautotrophs. They use sunlight, right? You can also have chemoautotrophs, which use chemicals. So for example, if you have a deep sea bacteria lives beside a volcanic vent, they can often use the sulfur that's generated by the vent for their making their food, okay, making energy. And so that would be chemoautotrophs. Okay. And then we have your typical parasite, which a lot of it is your decomposers. This is why things break down in this world and we can recycle nutrients, because a lot of prokaryotes will digest and break down molecules into smaller basic functional units, and then we can reuse them to build other things. Okay, so based on the way they feed. We can also classify them based on respiration. So this respiration is not breathing. What are we referring to when we're talking about respiration? It's something that we talked about when we talk about cells, things that they do. What do cells need in order to survive? Energy, right. So what's the process of making energy? Cellular respiration, exactly. That's what we're talking about respiration. Okay, so not the breathing in, taking oxygen is really because we need to do cellular respiration, right? So you have organisms that need oxygen and can only survive in oxygen-rich environments called aerobes, okay? And then anaerobes are organisms that cannot survive in an environment with any oxygen. So in fact, they will actually get harmed, okay? And then you also have organisms that can survive it with or without in the presence, okay, of oxygen. And so those are the facultative anaerobes. Okay, so they don't need oxygen, but they're not going to get harmed if there is oxygen present in the environment. So again, that can help us classify just based on what they need in terms of cellular respiration. Reproduction. How do bacteria reproduce? We talked about different means of asexual reproduction. What would be one way? Yes?
Binary fission?
Absolutely, binary fission. Okay, so basically allowing themselves to duplicate their genetic material and then splitting into two, right? So a very efficient and fast way to reproduce if resources are plentiful, and they can go through their reproductive cycles pretty quickly and really increase the colony size. And hopefully we'll see that when we do our culture plates. Okay, so what are some things that bacterial cells would need in order to reproduce? Yeah.
Food?
Food. So what do you think we're going to give? What kind of food are we going to give the bacteria? Think about cellular respiration. What are the reactants for
Sugar.
cellular respiration? Sugar. That's right. So we're going to give it some carbohydrates, and this is why at the bottom of the plate we have a little bit of agar, made up mainly with seaweed. Okay, and so that's going to be our nutrient for the bacteria. Okay, this is why bacteria they don't reproduce like on the table, like you don't see colonies forming all the time. It's because like, it's actually very limited resources just in the environment in general. So you have to kind of provide stuff for them to start. Okay, so you have your food. What else would help their growth? Carbon dioxide for why carbon dioxide in particular? What would you like the bacteria to use that for? That's okay. So somebody else said carbon dioxide. Yeah.
Humidity.
Humidity. And why is that?
So like it's a more moist environment. It's a little bit warmer.
Oh, okay. So not only humidity but temperature. Okay. So how will we increase the temperature for reproduction?
Maybe like put it out in the sun.
Yeah, absolutely, we can. But it's sunny now, but next week or over the weekend, we'll get rainy and cold. Yeah.
We can try controlling the temperature by putting it in an incubator.
Yeah, we can give it a little heat oven, right? So that's what we're gonna do. Is we're gonna temperature control it. But what temperature do you think bacteria can grow really good at? Around that.
40 degrees.
We can do a test run after we have 2 incubators. Let's try it. You guys pick 2 temperatures and we'll put them in there and see which one grows better. We can test our hypothesis. So 40 degrees one of them. Okay.
That's really
Order basis for that. Okay. We can try. Yeah. Okay. So we have temperature. We have food. Okay. And then what is the last thing we need?
Carbon dioxide.
Why do we need carbon dioxide?
For cellular respiration.
Do we need carbon dioxide for cellular respiration? Is that one of the reactants? No. We need oxygen. That's right. We need oxygen. Absolutely. So, if they are aerobic bacteria, they will need oxygen. If not, then probably won't. So yeah, there's space in here, so there's going to be access to oxygen for sure. Okay. Anything else? So if I put this into the incubator right now, would it grow? Why not? You just said you have everything we need.
We need to swab it and then.
We need to actually go collect some bacteria, right? So yeah, we need to put bacteria on here. So I'll talk a little bit about technical stuff. Okay. So yeah, most of this is going to be asexual reproduction through binary fission, and then hopefully we'll leave it in over the weekend and see if there's any. Okay. But bacteria can also go through sexual reproduction. So where they're changing DNA, and this is really beneficial for them if they want to pick up new genes that perhaps can help, them become antibiotic resistant, or there's some benefit trait. So they can actually build a bridge between 2 bacterial cells and transfer their circular DNA, which are called plasmids, over with particular genes that they can, the host and or the new bacterial cell can incorporate into their genome very easily. So their genome is more easily manipulated than the human genome. Okay, so that is for bacteria. There's one other group of prokaryotes. They've been classified as bacteria for a really long time, called Archaea, and they look very similar. One of the key characteristics is that we found that Archaea can live in pretty extreme environments. So very hot temperature, very salty seawater. In areas that are normally pretty harsh conditions, you can often find these prokaryotes there. Okay, and they also have some differences in terms of no peptoglycan in their cell wall. So that's one of the big differentiating factors that made us remove them from the bacteria classification and make their own group, is because they don't have peptoglycan like eukaryotic cells don't have peptoglycan in their cell walls. Okay. And but otherwise they do look very similar to bacteria. And then why this is important is because there is some suggestion that perhaps there was a common ancestor between Archaea and Eukarya. So these are your current animal, plant cells, protists that we'll be talking about. Is that they maybe had a common ancestor back where the common ancestor lost the ability to use peptide as part of the cell wall. And so they're actually more related to eukaryotic cells, even though they don't have a whole lot of organelles like from here. All right, so what are we going to do for in terms of keeping track of where you're going to collect your bacteria? So you have four quadrants on your plate. So you're going to be working with your partner. Okay. One of them we're going to keep blank. Why do I want to keep this one blank?
Control.
Why do I need a control?
So we,
You're right. I'm going to keep it as a control, but why would I leave it blank and put no bacteria on this? Yes.
Make sure it's actually bacteria that's growing and not something else on it when you put in all the other like food on it.
Yeah. So it may be a right reference, right? So if maybe the agar itself had bacteria. Who knows? Right? So it's just like a reference for comparison. Okay. So you know that you've actually gathered bacteria maybe from that exact location, and it's not just maybe the contaminants that were in the agar or other part of the petri dish that's going to actually grow. Okay, and when you go out and collect, sometimes you collect more than just bacteria. Like fungus will also grow very easily on here. So we'll hopefully get a chance to kind of compare those two. Okay, so you have 4 quadrants. You're going to leave one blank. You're going to pick 3 other locations that you think might have bacterial growth, and you're going to go collect it. And we're going to use swabs. Oh, I forgot to wrap gloves. Whoever is doing the swabbing needs to wear gloves because we have bacteria here, right? So we don't want to transfer that onto your plate unless you do want to grow bacteria on your hand, which you're welcome to. Okay. So grab some gloves. Can you please make sure that you reuse the gloves? You just want to like put your name on it at the end. You're going to store it on the side because in a few days we'll do like dissections and. Okay, so in terms of how we're going to do this, so here's my petri dish. There's a cap on top of the dish, and you'll notice that the little cross thing is on in the minutes. Can I just finish and then you guys can decide where you want to go? I know you're getting excited, but okay. So on the bottom there's a cross, and this is the part where the agar is on the bottom plate, okay? And so what you're going to do is when you're trying to collect bacteria, you don't want to leave this wide open because there's going to be airborne bacteria. You don't want it to get contaminated. So you're going to keep it as closed as possible, as often as possible, okay? And when you're swabbing it, you want to make sure that you just open it up like slightly. So turn the plate, give your partner the corner that they're supposed to swab, and just like open it like this so it's not completely like wide open. And then when you're swabbing, you need to make sure that you're wearing gloves when you grab your cotton ball. And then we want to make sure that we are very gentle. Treat this like Jello. It will crack. If it cracks, bacteria is not going to grow. Okay, so don't press too hard. And so you're going to take your swab and just gently kind of go like this, back and forth motion onto the plate, like that. So make sure that you do, like, for example, if I'm trying to collect bacteria from this counter, I want to make sure that I make good contact with my cotton swab, and then swab it on here. And I'm going to do it at 3 different locations. Please make sure you write it down, okay? And then we need to label this, right? So let's say location number 1, let's do the doorknob of the classroom. So you're going to write 1 on your petri dish, and I would either do it like just like a tiny number in the corner, because you don't want to do a big number. It's going to obstruct your view when you're looking at it under the microscope. Okay, so just do a tiny number, so it's a little simple, so you know what quadrant is 1, 2, 3, because those are 3 locations that you're going to pick from. Okay, you're going to swab it, and then you go to your next location, do the same thing again. Please use a fresh one every single time. Okay, and if you are doing a surface, you want to make sure that you don't just swab the same place over and over, right? Make sure that you get a good area. And then once you have your 3, you're going to come back. You're going to seal it up, okay? We're not gonna open this again because the chances are we might be collecting some pathogenic ones and we don't want people to get sick. So once you've done your collecting, we're gonna completely tape it up. And then when you do your observation, you're not allowed to open it again, okay? There are 2 places I'm gonna ask you not to go to. Number 1 is the bathroom. Why is that? Because it's, there's a lot higher chance of getting pathogenic bacteria, and we don't want to grow them in large numbers, okay? The other one is the cafeteria. And the reason for that is just because there is going to be bacteria at the cafeteria, we know that, okay? It's not necessarily bad bacteria, but we just don't want people to be, oh my god, that's from bacteria, and then, somebody reports it to the school board and have to get an investigation. So, like, it's just not necessary, because bacteria is going to be everywhere, and especially when you give it food and energy, it's going to grow, right? So it's not necessarily going to be, disease-causing agents, but yeah, just avoid the food selling places in general, okay? But anywhere else you can think about collecting. Okay. And let's all try to do different spots, because then we get a good spread. So, any questions before you go out? Yeah. I need you to choose one person to be a lab partner. Okay, I'm going to try to do this in pairs. And so,
swap this chance remote. Oh my god, yeah. Wait, I feel like
once you have your partner, come on up.